So today, yeah, wow, so many fewer people here today. Go figure. Today we're going to talk about the last set of RNA viruses for the term, with the possible exception of the very last day. I'm still trying to figure out if we can get a guest lecture for that day or not. Maybe we'll talk about some of the Bunya viruses that we haven't had a chance to talk about so far. Anybody see the article, I think it was in the Oregonian, about, I know it's NSF, where faculty members at Washington State University in Pullman think that growing bamboo may cause more hantavirus outbreaks. It's actually really kind of an interesting story. So if we get Dr. Wadas to come and speak about that, he'll talk more about that. No, again, I don't have your Scantrons, but the high score was 44. But again, that was a slight outlier, so we'll probably just go with normalizing to 42 again. A couple people sent me emails about the percentages you get in D2L. Ignore those percentages. It has absolutely nothing to do with what your final score is going to look like. What I do is I take the, what I normalize to, I'll add all the exam scores up at the end, I'll look at that, then I will calculate from there what the 100% is, and then go from there. So, and again, 90% are your exams, 10% are coming from the clickers, which will also be normalized to whatever that high score is. Okay, questions about that at this point? No. Okay, good. So today, this is my version, everyone's classic version of what kinds of different viruses, what kinds of different virus replicons you have. This is slightly different than what's in the PowerPoint online because I put it there this morning, but I'll repost this one. We're going to be talking about these viruses, the Baltimore class 6. Who cares about Baltimore class 6? It's the retroviruses. So in order to get to the messenger RNA, which of course is critical because these, like all viruses, are using the cellular translational machinery, these guys go to a single-stranded DNA, which is this black line here, then to double-stranded DNA, and then finally to messenger RNA, which is a really bizarre mechanism in terms of how to replicate first your genome, and then also in terms of replicating your virus, but, the virion I should say, but the main reason this seems to be happening is because these guys can now integrate into the host genome, and at least for the retroviruses, it's absolutely required that they integrate into the host genome. And that is a great way to keep your genome maintained, because your genome is now in the host genome. And so whenever that host replicates, your viral genome is being replicated as well. So again, retroviruses, these are the single-stranded DNA viruses with a double-stranded DNA intermediate. I should have added that there. It's not just the ones that integrate into the genome. There are a few viruses, in fact, the viruses that I had in the review on Monday. Those also replicate through these RNA intermediates, DNA, what's being packaged, etc. The main reason that people were excited about these particular viruses and the reason they found them in the first place was RNA tumor viruses. And no, we're not going to go through the whole book. But this was really surprising because cancer is a genetic disease, right? And what are our genomes? They're DNA genomes. So how can a RNA virus be causing tumors? And so that's how these retroviruses were found in the first place, was by studying these RNA tumor viruses and figuring out how that replication process took place and the DNA. It's also in the study of these RNA tumor viruses that oncogenes were originally discovered. And it was these RNA viruses which had picked up cellular genes which were important for replication and the control of replication of the cells. Now, this is way above and beyond the replication of the virus. Of course, the most famous of these is HIV. We'll talk about that towards the end of today's lecture, and then maybe also a little bit on Monday as well. The structure of HIV has this really, well, particularly the nucleocapsid core of HIV has this really fascinating structure called a fullerene cone. What are our key concepts? Far too many of them here, but mostly has to do with how these guys replicate. Again, this really bizarre mechanism whereby it goes from RNA to DNA and then integrates into the genome. The way that it replicates, just like all DNA polymerases, these guys need primers, but these particular DNA polymerases use a cellular tRNA as a primer, which is a really interesting and it's a great mechanism because, again, it's there, something that all cells have to have, so antivirus can actually take advantage of that. They use frameshifting, in fact, a lot like we've seen in a number of different viruses, the coronaviruses, et cetera. HIV in particular uses splicing to a great degree, but it turns out that all of the retroviruses use splicing to pretty much make some of their very specific genes. Reverse transcription, of course, we'll go into in great detail. The reverse transcriptase, of course, is the molecule which does this. Once you've done this reverse transcription, then you've got DNA that needs to get integrated into the genome. And because of the mechanism of how this reverse transcription takes place, you end up with these things called long terminal repeats. And these long terminal repeats are really critical for making the RNA, which is your genome, and also the messenger RNA that gets made into virions and moving along. Part of the process of reverse transcription and generating these LTRs is something called template switching. So we talked a little bit about recombination again with the coronaviruses, where they're probably skipping from one part of the genome to the other to make the subgenomic RNAs, and probably also in terms of helping to repair deleterious mutations. It's one of the questions on the exam. I'll bring those exam questions on Monday when I have a chance to analyze them. So that's something which has to happen in terms of retrovirus replication, as opposed to probably happens in other virus replications as well. And then the last thing I wanted to mention here are the horrible naming structure that these guys have, GAG, POL, and ENV. A lot of that has to do with the discovery of these RNA tumor viruses. The GAG are the group-specific antigens. They're basically the structural proteins. So we talk most of these when we talk about the structural proteins. The non-structural proteins here are all of the POL proteins, so it's not just the polymerase. And you call it polymerase even though it's a reverse transcriptase. Again, not very good naming structures. And then ENV is the only one that actually makes sense. These are the envelope proteins which are present there. So again, it's typical for our outlines. We'll talk a little bit about origin of diseases in general for retroviruses. I wasn't planning on talking very much about HIV disease. There's plenty of stuff out there about that, which we won't get into. Structure, and really importantly, not so much the structure of the individual virions, which are shown over here. This is some of the budding processes of each of these individual virions. Here are virions on the inside. It's a little hard to see with this projector, but they also have these nucleocapsid and these enveloped virions here. Entry is pretty standard, receptor-mediated, conformational changes. We've even seen that already. Big difference, of course, is replication. Transcription is pretty straightforward, as is assembly, although there's some interesting differences there. So retroviruses. Almost everybody thinks about this one right here, HIV-1. But this was by no means the first retrovirus to be found. In fact, it was true for many viruses. It was a disease, and then they found a virus that was associated with it. The original ones were these, the sarcoma viruses, which were, again, RNA tumor viruses, where they were originally found. Alpha, beta, gamma, delta, epsilon, not a big deal. Important here in terms of these are the lentiviruses. Why are they called lentiviruses? What's a lenti? Slow. So these are the slow replicating retroviruses. HIV, SIV, FIV, etc. And the spumaviruses are actually rather quite similar to these guys as well in terms of being very slow. All of these have reverse transcriptases, and at least all of these up here, sarcoma, mammary tumor, leukemia, dermal sarcoma, are oncogene-containing viruses. What does a general retrovirus structure look like? we'll start from the inside and go out. They've got 2 copies of their RNA genome, which is packaged inside. They're more or less identical to each other. So it's not like what we had in the case of flu, the orthomyxoviruses, where there are lots of different segments which are there. They're just 2 basically identical. The differences they have is due to the differences in replication, the problems that they have with replication. As each of these genomes are present in here, they also have a tRNA. This is a cellular tRNA, which is packaged here together with this, well, these 2 copies of the genome. Also have, now not terribly surprisingly, a nucleocapsid protein. What's a little bit different here relative to some of the other viruses, there's also a capsid protein. So they're 2 separate things. The nucleocapsid protein is binding to the RNA. This capsid protein is making up the internal structure which is present on the inside of these virions. Here we also have the reverse transcriptase and the integrase, all packaged as proteins inside the virion. And that's because, just like we had for all those negative strand RNA viruses, those need to have proteins in order to make something which can be making messenger RNA. The only way these guys make messenger RNA is once they've integrated into the genome. So you have to reverse transcribe them, you've got to integrate them before anything is actually going to happen. So you've got to bring all of this protein machinery with you. You also, of course, have to get inside the cell. That's through these 2 proteins, usually go by transmembrane and surface protein, TM and SU. Again, function a lot like our HA1, HA2, etc., all the stuff we've talked about in terms of influenza virus. There's also a matrix protein here that sits right underneath the membrane, which is important for putting all of these things together and, in fact, connecting this envelope here to the nucleocapsid on the outside, really holding the whole thing together. What do these genomes look like? This is, again, a very generalized retrovirus genome, what's being packaged inside each of those particles. Again, there's 2 copies of this. They've got a cap. They've got a tail. Where do those caps and tails come from? Cellular, because this is replicating in the nucleus. This is totally normal cellular replication, transcription, which is going on there. So the cap and the tail come from normal poly-A polymerase. The cap comes from the normal capping enzymes. On the other hand, the genome itself, this is the part which is specific again to the viruses, has this repeated sequence R, because it's the same sequence at the 5' end and at the 3' end. Then there's some unique sequence at the 5' end. It's the U5. Some unique sequences at the 3' end. There's a primer binding site and a polypurine tract. So these are just the sequences here. Major splice site, 5' splice site, and a 3' splice site. So what happens, again, normal cellular splicing processes. If you look at what happens with this splicing, if you splice from here to here, you're going to end up with a lot of the SU and TM proteins. These are the envelope genes. This was going to go on the outside. If you don't do splicing, then you also end up with the GAG proteins. So GAG are the group-specific antigen. Again, it's the groups of different RNA tumor viruses here. But they're the main structural proteins. It's the matrix protein, the nucleocapsid protein, and the capsid protein. You'll notice here that there's a slightly overlapping open reading frame here for the Pol genes, multiple genes here, reverse transcriptase and integrase. The only one we haven't talked about so far is this guy right here. Find in lots and lots of positive strand RNA viruses that make polyproteins. It's a protease. So matrix, nucleocapsid, capsid, protease, reverse transcriptase, integrase, surface, transmembrane protein. All retroviruses have these. HIV has some slight differences there. How do these guys replicate? Again, this is shout out to the appendix of the Flint textbook. Bind to receptors on the outside of the cell. These guys fuse at the plasma membrane, so it's not a pH-dependent change that happens in the endosome. Your capsid is released. That capsid has the 2 genomes, all of the enzymes that it needs. The reverse transcriptase is present here. It starts doing its reverse transcription thing in the capsid once it comes in. You end up with a double-stranded piece of DNA here in the cytoplasm, again, through the activity of the reverse transcriptase. This double-stranded DNA, together with the integrase protein, gets into the nucleus, integrates its genome into the nucleus. Then you have cellular transcription, some of which leads to your structural proteins, GAG, again, the major structural proteins, some of which is this long protein, which also has the Pol protein. You also need to make your envelope proteins. That's that splicing event. Here, RNA gets assembled in the normal secreted surface protein process. All these guys come together at the membrane. You bud off, and you make mature virus particles. Yeah?
If it's not a pH change, what's activating the reverse transcriptase?
So what activates the reverse transcriptase inside the capsid once it gets inside the cell? It seems to be just the presence of DNTPs, which allows it to function. These guys were originally found. The reverse transcriptase was found independently. Whether it's independent or not is not entirely clear, by David Baltimore's lab and Howard Temin's lab, published at the same time in Nature. They just purified virions and looked at activity. So it was just purifying the virions, throw that together with your radiolabeled DNTPs, and it works. So it's active just in and of itself in the virion. What does it do? Well, okay, so this is the molecular gymnastics is one term for it. Convoluted is another way of looking at it. How this process works in terms of getting replication to take place. Again, rather baroque gymnastics, etc. So again, we've got our positive strand genome up here at the top with repeated sequence at both ends, 5' end, 3' end, positive strand RNA, right? What direction do polymerases go? 5' to 3', right? So you have this primer binding site. You remember the PBS. This is present in all of these retroviral genomes. Binds to part of a tRNA. That tRNA is providing what? It's a primer, right? So primers have what? 3'OH, exactly. So the 3'OH is here, primes your reverse transcriptase, copies out to here. That's a silly place to have a primer binding site, isn't it? Why have it all the way out at the 5' end? You should have it way over here, right? Okay, we'll see why, actually, evolutionarily, this is how it happens. So, yes, copies to the 5' end of the genome, which is actually not very far. It's a relatively short piece. What it does then is it gives you a complementary copy of this repeated sequence right here. What happens when you've got an RNA-DNA hybrid? Gets chewed up by RNase H. Normally there's a cellular RNase H. In the case of most retroviruses, the polymerase also has an RNase H, say the polymerase / reverse transcriptase, also has RNase H activity. So it chops up the RNA, which is here in this RNA-DNA duplex. Now you've got this complementary sequence to your repeated sequence here at the complement of what was the 5' end of your genome. Well, now you've got a repeated sequence over here at the 3' end. So this is what's called the first strand transfer. This is that first recombination. As I mentioned before, you've got to do this recombination strand switching for replication in all of these retroviruses. So now you take your primer together with this short piece of DNA, and pop it over to now the 3' end of your genome. Now this is great. Look, we've got this 3' OH that we had from over here. We've got this whole genome in front of us. Now we can make a copy of this all the way out to the end of the primer binding site because the rest of this got chewed up before. So now what we've done is we've taken this unique sequence that was at the 5' end of the genome and made a complementary copy of it over here at the 3' end of the genome. We've copied everything here. Now we've got RNase H that chews up everything here, chews everything up, but we still need to make that second strand, right? What do you need to make this? You need a primer. Well, it turns out that the primer here is this PPT, which is resistant to getting degraded by RNase H, and it serves as a primer as well. But this is over here at the 3' end of the genome. So it replicates over the end here, and basically it should be becoming really obvious what happens next. These guys get chewed up. The tRNA gets chewed up. Now we have a complementary sequence at this end to this end. This sequence jumps over to the opposite side. This is our second recombination. Now we replicate our way down to the end of the genome. We replicate our way down to the end of the genome. And what's happened here is now we have this sequence that used to be at the 3' end, repeat sequence, which has always been at the ends, and 5' sequence and primer binding site. But here we also now have this unique sequence at the 3' end, but the unique sequence at the 5' end. It's because of this template switching that we end up with these repeated sequences at either end of the genome. These are known as the LTRs, or long terminal repeats. Turns out that long terminal repeats are among the best promoters for RNA polymerase II that anybody has been yet able to find. They're absolutely fabulous promoters. Yeah?
So I have you know how it extends to make the PBS?
Mm-hmm. Yeah.
What is that making it out of?
Okay, so where does this PBS come from? That's the part of the tRNA right here, which is bound way back over here. So that part of the tRNA is complementary to the primer binding site because the tRNA is the primer. Yeah?
When you've made all this, how do you then go back to making it back into RNA for packaging?
How do you get it? So we'll talk about that a little bit later. But it turns out that, yes, these LTRs, the promoter is right next to this repeated sequence. And so that's where you start to make the messenger RNA. And then the terminator for transcription is right next to this other R sequence.
So then how do you get the PBS and the PPT sequences back on there if you chew them off?
You chewed them off, but then you made them again.
Oh, are they still in there? They just weren't marked.
Yeah, well, so PPT, so this is what you start with. PPT is here. Right, and here's copying. It's just making a copy. So that's what's coming. Yeah, in the back.
On step 5,
Yes.
you completely degraded the complementary RNA strand and you're running the DNA. So does reverse transcriptase transcribe the complementary DNA strand, or is that DNA?
Okay, so here, yeah, we're going to talk about that in just a second. But yeah, so unfortunate here, everything here I should have flipped it the other way around. So black here is RNA and brown is DNA. So 5 here is identical. I just copied and pasted this because it's too long for one slide. So the RNase H here is degrading most of the RNA, but now this PPT is serving as a primer for not the DNA polymerase. You remember, all this is happening in the cytoplasm. Is there any DNA polymerase in the cytoplasm? It's all in the nucleus. So it's all the reverse transcriptase which is doing this. So reverse transcriptase is really kind of a Swiss Army knife in terms of polymerases. It's an RNA-dependent DNA polymerase, it's a DNA-dependent DNA polymerase, and also has this RNase H activity. So really pretty amazing protein. But one of the things it doesn't have is proofreading. So it's actually really quite a low-fidelity polymerase. And this is one of the reasons why HIV drugs have been a real problem to make and get to work. And that is that you end up making lots of mismatches, mistakes when these genomes get replicated. And what you end up with for all of these retroviruses is what's called a quasi-species. We haven't talked about quasi-species much yet, but it's true for all of the RNA viruses that are replicating, again, through these RNA-dependent RNA polymerases or even a RNA-dependent DNA polymerase. You end up with a whole mixture of different sequences. And so whenever you do a virus infection, you've got, of course, lots and lots of virions. Almost all of those are going to have slightly different sequences relative to each other. And it makes, again, antivirals really problematic because there's going to be a very large population, some of which are going to be mutant. And you select against one of them, but then some of the other ones will still make it through. Basically, I already talked about this already. Reverse transcriptase, there's lots of copies of it in the virion, as well as being a not proofreading enzyme. It's also not very processive, so it falls off the template all the time. Does function in virion preparation. We talked about that before. Basically has 4 activities. An RNA-dependent DNA polymerase, as you're asking about, was a DNA-dependent DNA polymerase. Also has a DNA helicase activity. It can separate the 2 strands. And an RNase H. This is a specific domain of the reverse transcriptase. That reverse transcriptase looks like this. Again, it's kind of hard to see here, but it's got a palm and thumb and fingers domain, which should sound really familiar relative to poliovirus and all of the other DNA polymerases. Here, what's different is that we have an RNase H domain. And so the RNase H, the RNase, which is going to cleave up DNA when it's in a RNA-DNA hybrid, is part of the reverse transcriptase. So it comes along and is part of that process as it comes together. And again, the structures here look extremely similar to each other. The poliovirus RNA-dependent RNA polymerase, the HIV-1 reverse transcriptase, and DNA polymerase 1, 2, 3, you name it. They've all got very similar structures. So now we've made our double-stranded DNA. It's got these extra LTRs at the end. Now we need to put this into the genome. How do we do that? It's through the integrase protein. Integrase protein binds to these sequences at the end of the genome. Again, these are the repeated sequences, long terminal repeats. U5 are, that's U3RU5, U3RU5. They have these specific sequences. The actual sequences here are not important. The integrase binds to these, cuts slightly offset in the genome, just like what happens with transposons jumping into the genome. Then you have gaps. These gaps get repaired by cellular polymerases. You end up losing a couple of nucleotides at the end of your genome, but do you care? No, because the only part that you actually need in terms of the virion is what's beyond these repeat sequences. So those extra sequences are really kind of immaterial. Yeah?
On here and on the other side, are the repeats, are the R's just because the repeat sequences, or is that referring to them being like purines?
Okay, so what does the R stand for here? The R just stands for repeat. It has nothing to do with, large amounts of purines, pyrimidines, et cetera. So it's to say literally a repeated sequence. There's a decent video for integration here. It's actually really crummy in terms of replication as far as I'm concerned. So, but looking at integration, talks about how the ends are cleaved here. They get put back together. And you end up with these direct repeat sequences on either end of your integrated viral genome. And the reason for that is the offset cut, which happens here. So here you cut in a different place. You hook up those 2 ends together. You end up with a gap here. That gap, again, gets filled by the cellular repair processes. And then in between these directly repeated sequences, you have the viral DNA. And this is now called a proviral DNA because it's been integrated into the genome. Yeah?
Do these integrate at a certain place in the host genome, or is it just anywhere?
So up here, we have random,
Big word there.
but it's in quotes. And so it's thought that integration is more or less random. And so that's why, thank you for pointing that out. So it's not GGGCCC. where it always integrates. It seems to be actually more in AT-rich parts of the genome, but probably almost as important as that sequence is the structure of the chromosome at that particular place. So if you've got heterochromatin, which is heavily compacted, you don't have much integration that happens there. You have euchromatin, which is not compacted, you have more integration there. Makes perfect sense. The problem is when you try and use these kinds of things for gene therapy, then they're going to jump into parts of the genome which are being actively transcribed because they're euchromatin. So you may have heard the story about the poor bubble baby, one of the very first gene therapy trials where the guy died of leukemia very soon thereafter because of this gene therapy integration and causing activation of genes. Maybe that's too far away. But yeah, no, it turns out it's not completely random. People used to think it was random. If you look at it in a test tube, yes, purified DNA, purified integrase, it does jump in at random. But if you think about genomes, it's actually not completely random. But more of it has to do with euchromatin versus heterochromatin. Where's the easiest place to jump into? Exactly. Okay, now you're in. This is great, but we still haven't made any of our genome. We haven't made any of the proteins. How do we get those? That's through cellular transcription. And again, these LTRs are fabulous promoters, and the place you start is immediately 5' to this repeat sequence, and where you stop is immediately 3' to this repeat sequence. There's actually one question that should immediately be coming up here, which is, if it's a repeated sequence, isn't there also a termination site right here? And the answer is yes, there is, because it's a repeated sequence. But there's suppression of termination, and we don't have time to get into it. It's very well described in the Flint textbook if you're interested in looking at that. Again, cellular capping, cellular tailing. What do you get? You get basically two major kinds of RNA. One is the full-length RNA, which is from repeat here to repeat there. We also mentioned the splice sites. Then you have a spliced RNA, which gives you envelope proteins. Here, GAG Pol is made as a polyprotein, where you've got GAG protein together with your polymerase protein, which of course means you need a protease to chop these guys apart. How do you get that polyprotein? Turns out that the Pol proteins, as I mentioned right at the beginning, are not in the same open reading frame as the GAG proteins. You have to have a frame shift during translation. That works in two different ways. Turns out that there is not just termination suppression for transcription, but also for translation. Secondary structures form in the RNA. Turns out it's different for different kinds of retroviruses. Here it's just known as a stem loop structure. So this big gray blob is supposed to be your ribosome, happily translating its way along the viral RNA, gets to this secondary structure, gets bumped back one nucleotide, and then continues in a different open reading frame. Here, the open reading frame would continue UCC, AUA. Here it's now CUC, CAU, AGC, etc. So reads through this otherwise termination codon right here. And again, it's all due to the secondary structure. What this means is that sometimes you get translational termination and it falls off. Between 5 and 10% of the time, you get suppression of termination and you make the polyprotein. So you have a lot more of the GAG protein than you have of this polyprotein which is being made. How do you go from polyprotein to actual structure? Again, it's through the activity of the protease, again, which is right here in the GAG-Pol polyprotein. And just like the proteases we've talked about for all of these other positive strand RNA viruses, it works in the polyprotein. So it cleaves up the reverse transcriptase and the integrase. Also will cut matrix nucleocapsid and capsid apart from each other. What's interesting is if you look at this structure, matrix, nucleocapsid, capsid, I should have brought up the cartoon of the virion. That's also exactly the way they're arranged inside the virion. So if you go from the outside to the inside of the virion, first you have matrix, then you have capsid, then you have nucleocapsid. So all three of those are right together. And it turns out the way this works, and in some previous years, if you go and look at some of the older notes, I had Dr. Barkless from OHSU came down and gave a guest lecture. He does assembly of HIV and other viruses as well, but particularly HIV. And so his lab and some other labs have shown really nicely that what happens here is it's not a coincidence that it's like this, matrix followed by nucleocapsid followed by capsid, is that this matrix protein gets stuck into the membrane, and then you have assembly of the virus particle right there, and then the protease actually chops these into smaller pieces really late, and in some cases actually even after the virus is budded off of the virion is budded off of the cell. You also need to package your genome. How do you get genome packaging? It's through this sequence right here, which I haven't talked about yet. This is the Psi sequence. It's in that RNA. It's what gets bound to the nucleocapsid protein, which will take that and make it into your final piece that goes into the center of your virion. This Psi sequence, you'll notice, is downstream of the 5' splice site, and that means that you only package an RNA which has not been spliced. If you're packaging RNA that was spliced, then you'd have these really tiny RNAs which would not be functional. You have to then splice these ones that are here. And again, the protease is active only at the very end of the process. So that's general reverse transcription retroviruses. Where do you get all these RNA tumor viruses from? Well, it turns out that the problem with these RNA tumor viruses is that they have integrated into active parts of the genome. Again, not terribly surprising. Where do you have active parts of the genome? Well, that's very often where you have genes that are being highly expressed, say for cell division, replication, etc. So it's not terribly surprising that some of these retroviruses have been integrated into the genome right next to proteins which are really important for cellular proliferation. And it turns out that the termination that normally will happen from a long terminal repeat over here is not always that good. And so quite often you'll end up with an RNA which is a little bit longer than it would normally be, and it picks up some of these protein genes right next to it. And many of those genes are oncogenes, and that's how oncogenes were first discovered and how we got RNA tumor viruses, because of the integration into the genome. These are just two different kinds. Rouse sarcoma virus actually has the whole genome in it, together with a oncogene. There's some other viruses which don't have envelope proteins but have picked up these oncogenes as well in terms of expressing them. As well as these RNA tumor viruses, which fortunately are relatively rare. There are also large numbers of proviruses that are present in our genome. And this is just a figure from the molecular biology textbook looking at a number of these retroelements which are present in the human genome. So there are on the order of 8% of our genome are these endogenous retroviruses which have everything in them that they would need to make a virus. They've got 2 LTRs, they've got GAG, they've got POL, they've got ENV, and just again as a quick reminder, 1.5% of our genome is the protein coding sequences, the non-repeated protein coding sequences, so we're more viral than we are human. And I talked about this a little bit at the neuroscience meeting, what was it, Wednesday after the midterm? Feels like years ago. So there are a number of these what are called human endogenous retroviruses, HERVs. At least 1 and probably 2 of them are really critical for placental development. So without these HERVs, we wouldn't have Mother's Day and be here at all. So very good things, at least as far as I'm concerned. There are a number of what are called retrotransposons in the genome, which just look like these endogenous retroviruses, We're just missing the envelope proteins, or protein genes. And there are, again, very large numbers of these in our genome, really large amounts that are there. Then there are also these very bizarre other things that we have in our genome. They're called line elements and sign elements. So long interspersed nuclear elements and short interspersed nuclear elements. These guys have direct repeats at either end of them, and they look as if they were at one point a messenger RNA, because they've got these poly A tails at the end of them, and most of them actually include, particularly for these line elements, they've got reverse transcriptase genes that are encoded in the middle of them. How people think these work is here. We have a long interspersed nuclear element in the genome that gets transcribed. Poly A tail, again, normal cellular processing. That gets exported into the cytoplasm. In the cytoplasm, there are in fact two genes which are encoded for in this line element. This is one of them, it's an endonuclease. This one's a reverse transcriptase. These guys bind to this messenger RNA in the cytoplasm. That gets imported back into the nucleus together with the reverse transcriptase. It binds to a sequence in the genome where there are a number of Ts. Why a number of Ts? Because we had a poly A tail that was made in this messenger RNA in the first place. Then there's also the endonuclease activity, which will cut the genome right here. It uses these Ts now as a template to make a DNA copy. That DNA copy then gets integrated into the genome. This happens many, many times, and we end up with almost 40% of our genome, which is lines or sign elements. So massive numbers of these things, which, again, I think are related to retroviruses. People will argue about that. It's also another really cool mechanism about viruses, but we won't talk about that now. Okay, any general questions about retroviruses? Otherwise, we'll talk about some of the interesting twists that HIV takes on the molecular aspects of replication here. So, HIV-1, yes, nasty disease. Probably, again, one of those which is way up there with influenza, dengue, in terms of absolute numbers of infection. Fortunately, we've been able to lower the morbidity that's happening from HIV really quite well. How does this disease progression usually work? There's an acute infection that takes place, very high amounts of viral RNA and virus capsids, and then there seems to be a pretty quick decline in the amounts of viral RNA. And this is one of the big problems with getting rid of HIV. You can treat HIV, but it's very hard to get rid of. Incubation periods of years, and this varies a huge amount here. And then at some point, and it's not entirely clear how and when this happens, you have a real spike in terms of the amount of viral RNA and the amount of disease which happens, and this is when AIDS happens. And it's an acquired immunodeficiency syndrome. The virus doesn't kill you, it's all the other infections that do, because you've lost most of your immune response. And so that's what's down here at the bottom. This is what really is causing people to die. Usually, again, very low load that you have through most of this process. Seems that the NEF protein is really important for this. We'll talk about NEF in just a couple of minutes here. NEF mutants, these are HIVs that stay at this level basically forever. And so one of the really promising HIV vaccine candidates is one which is deleted in this NEF gene, and hopefully we'll be testing some of this here at PSU in the relatively near future. What happens, again, cytotoxic T cells. These are the killer T cells that would normally be helping out with disease. Those are the ones which get infected by HIV. And again, most of the time it's latent here. What are the structural proteins? They're very similar to what we've already talked about. matrix capsid, nucleocapsid, protease, reverse transcriptase integrase, SU and TM. The only other thing which is different between HIV-1 and all of these other retroviruses is this virion protein R, VPR. Unfortunately, there's a lot of other names that people use in HIV. You may have heard a lot about GP120. GP120 is the main surface protein. That's what's interacting with the receptors. Also, people will talk about GP41. The reason they've got all of these strange names is because that's the relative molecular mass of each of these things. So this one's 17 kilodaltons, 24 kilodaltons, 32, 120, 41, etc. Again, not very good naming procedures. The virion looks extremely similar, 2 copies of the RNA. The thing which is really different is the nucleocapsid structure here, which is present in the middle. That's really not an icosahedra, not a clear helix. It's actually sort of 2 of them put together. This is a paper from a couple of years ago in Nature where they really did a nice high-resolution structure here. Basically what this is it's 2 halves of an icosahedron kind of squished together at either end with a helical symmetry in between. So each of these greens here represents a pentamer. Each of the blues represents a hexamer. But it's fitting together in an extremely similar way to how we talked about all of our T equals 7, T equals 3, etc. And that's one end, however, has a different T number than the other end. But it's these 2 basically half icosahedra fitting together at either end. The genome that's present on the inside here has our standard GAG, POL, and F proteins, but then a bunch of extra ones. First, this NEF protein, which seems to be important for this latency. If you have NEF, then you escape latency. If you don't have NEF, then you stay in latency, and we'll see how that works in just a couple of minutes. Then you also have these other proteins which are present through alternative splicing. TAT and REV are the ones that are best understood how they work. TAT and REV are important for transcription and for nuclear export. VIF, VPR, and VPU are proteins which are much more important for the pathogenesis process. So, TAT and REV are way more interesting than these other ones here. How do you get HIV inside the cell? It's through binding to receptors, through that now GP120 protein. Why they keep switching back and forth? I much would prefer to call this now our SU protein. Surface protein binds to CD4, which is a classic cellular marker for cytotoxic T cells. Binds to CD4 through a lot of work on HIV. We found that CD4 binding by itself is insufficient. You also have to have binding to a so-called co-receptor, CCR5. In this particular case, there's CXCR4 as well that does this. What's really interesting is the way that these were found. Anybody know how these were found? They found people who were actually mutant in these particular genes who were then resistant to HIV. So these were partners with HIV positive. One stayed HIV negative basically for the whole time. They went back and looked. They found there were mutations in this particular gene. Just is really interesting. Doesn't seem to cause them much harm either, which is curious. So binding to CD4 and a co-receptor, CXCR4 or CCR5. Then you have the conformational change, which takes place and releases the capsid and nuclear capsid inside the cell. Again, all this fusion happens at the plasma membrane. In the case of HIV, you need this VPR protein for nuclear transport, and then some of the matrix protein is also important for that process. We have a bunch of non-structural proteins in HIV that you don't have in all of these other retroviruses. We already talked about NEF. Why it's called NEF? Because it's a negative effector. See why that is? Actually has to do with budding at the end. There's a regulator of expression of virion proteins and a transactivator of transcription. That's what all these names come from here. And unfortunately, there's a protein unique to HIV, VPU, oh, great, and a viral infectivity factor. These, again, are much less well known, and they're much more important for pathogenesis. TAT and REV, however, we know a lot about how these work. First, TAT, which is the transactivator of transcription. It is a RNA binding protein. So the TAT protein binds to a very specific structure in the RNA, which is being made from the provirus in the genome. This is the TAR RNA, so the TAT response element. If you don't have TAT around, then the RNA polymerase II falls off of the provirus. You don't end up with any virus replication. In the presence of TAT, you have this binding together with some cellular proteins. Not important what they are, but they're listed over here. TAT binds. Now you have anti-termination. You replicate your way through the whole rest of the genome. So you end up with lots of messenger RNA. This is not a 100% process, because if it were a 100% process, you would never have TAT in the first place. Yeah?
So with the abbreviations versus the GP20, 120 and all that, do you want us to know the abbreviations?
I wouldn't know GP120 and GP41 because I keep forgetting them. So if I forget them, I don't think it's a reasonable thing for you to be expected to remember. And plus, it's much more important in general for all of these retroviruses. So TM and SU, yes, I think that's a reasonable thing to remember. But that SU in HIV is GP120. I'll try not to put that on an exam. I won't necessarily succeed, but I'll try not to. So that's what happens with TAT. And so at early stages, and again, probably this is what's keeping, one of the things which is keeping the virus at low levels through that whole latent, literally years when you have an HIV infection, it's because you don't have TAT around. So if you even did get transcriptional initiation, it would get to TAR, and then you would have dissociation. Only when you get this TAT protein are you going to get this full-length genome which is being made. There's also the REV protein. The REV protein is also made through alternative splicing down here at the 3' end of the genome. The REV protein is critical for getting RNA export from the nucleus. Again, all of this is in the provirus. So in the provirus, in the nucleus, you get transcription. That transcription is going to give you various different RNAs, particularly your first splicing, which is happening, again, normal 5' splice site right here, normal 3' splice site right here, envelope proteins, differential splicing in HIV will give you TAT and REV, which also have this splicing. These RNAs are made, they get exported to the cytoplasm. This REV protein is made, it's in a spliced RNA. That REV protein gets imported back into the nucleus. And then, when you have enough of the full-length RNA, again, we got lots of splicing going on here, right, in the viral genome. Normally, what happens to unspliced RNAs? Do they get out of the nucleus? No, they usually get processed. They don't have exon junction complexes. They can't get out of the nucleus. However, if you've got the REV protein there, it binds to a sequence in here, not surprising, that would normally get spliced out, and then allows this RNA to get transported into the cytoplasm, packaged inside the virion, and exported. So it's another way of making sure that you only get production of functional infectious viruses late in the process. So there's lots of controls. And again, you remember what's this group of viruses called that HIV is in? The lentiviruses. They're slow. They don't replicate very quickly. And so rather different than the other alpha, beta, gamma, delta, epsilon retroviruses. So it's another way of controlling that. These are the other non-structural proteins. VIF blocks the deoxycytidine deaminase. This is a viral protection antiviral protection mechanism. VPR helps get in. VPU degrades CD4. Why do you want to degrade CD4? It's actually something really similar to neuraminidase in influenza viruses. It's chopping off the thing on the outside of the cell that you'll be using to come in. So when you're leaving the cell and going to try and find another one, you don't want to try and reinfect the cell that you just came from. So this process, VPU, stops that. And NEF also seems to cause a decrease in CD4. The way that seems to work is you have CD4 that's either going to bind to VPU and get degraded. This gets degraded in the cell before it actually gets transported out to the exterior. Or if you have CD4 on the exterior, NEF will basically cause it to be sequestered and taken away from the surface of the cell. Now, why a NEF deletion should be so useful in terms of vaccine production, having CD4 around, you would think there'd be lots of infection, so I'm not quite sure why this actually works in terms of a vaccine, but it does seem to work reasonably well. So these are the processes, how you make the virus, how do you combat that, what are the current therapies which are being used in terms of treating HIV. This is the highly active antiretroviral therapy. I hate this abbreviation, but everyone calls it HAART all the time. So most of the time, I'm sorry, this has gotten chopped off down here at the bottom. There are 4 different drugs which are used. It's a cocktail of multiple different drugs which first inhibit the reverse transcriptase. They inhibit the integrase. They inhibit the protease. And they inhibit fusion and entry processes. So to start with, it was 3 drugs that were mostly used, and now in some cases we also have 4 drugs which are being used. The big problem originally with anti-HIV therapy is only one of these was being used, like a reverse transcriptase inhibitor or an integrase inhibitor or a protease inhibitor, because there's such a high mutation rate that happens in the replication of the genome, and because you have a quasi-species, a whole mixture of different genomes which are there. Very, very rapidly you ended up with reverse transcriptases, for instance, which were resistant to these different inhibitors which were being included there. Same thing was true with integrase inhibitors, same thing was true with protease inhibitors. But it appears that treating with 3, or in some cases 4 drugs really does shut down the ability of the virus to replicate, but you can't actually get rid of it. And so a lot of the current things that people are trying to do with anti-HIV therapy is really trying to get rid of those proviruses, really, which are integrated into genomes. And that's really, really hard to do without totally messing up the immune system as well. One of the treatments that people are talking about is doing full bone marrow transplants. And that does seem to work, but that's a really extreme way of dealing with disease. So that's it for the retroviruses. Again, I could beguile you with all of the nasty diseases for HIV, but I decided I wouldn't. And wish you hopefully a somewhat sunny weekend. We'll see. We can hope.